Intertidal lab, we're going to do a survey of the gradient from the upper intertidal to the lower intertidal. We're going to do a biodiversity survey here. The first thing that I'll do is I'll run this transect tape out 15 to 25 meters from the upper intertidal to the lower intertidal. And I'll try to go in a straight line, but it doesn't have to be exactly a straight line. And the rocks get quite slippery down here. About here, you can see the lower intertidal with the sea grasses and seaweed. So that's kind of where we're aiming for our last sample. But I'm not going to put the transect that far because I don't want it to get washed away by the waves. So of course this entire time that we're sampling, we're going to be very careful to watch the waves, not turn our back on them. So I'm standing at about the 10-meter mark of the transect tape in the middle intertidal. We would have measured 2 quadrats up at 0 meters and 5 meters. And now this third one is going to be at 10 meters. The purpose of the quadrat is that this is a known area of a ½ meter by ½ meter that we're doing the biodiversity observations here on the intertidal. So I put that down at about the 10-meter mark. And then what you're going to do with your quadrat is look in this area and estimate the number of species. There you can probably get a pretty accurate number of species, but your abundance measures you might have to estimate a bit, because if you look closer you can see there are all kinds of limpets, sea anemones. And if we look down into the low intertidal, you can see there's even more diversity of seaweed, and I see some snails just from looking in there. And we'll do 2 more quadrats down in that direction when the tide goes out a little further.